Professor George Malliaras. You used to watch those films where people are plugged into computers, and that all seemed a bit daft, really. It turns out that it might not be so daft. Professor Malliaras is working on this interface, dare I say, is a professor in the Department of Engineering, and is going to talk to us about what he might be able to do to us. Professor Malliaras, please.
Thank you very much, Sir Mike, for the kind introduction and the invitation. It's a pleasure to be here. I also would like to thank Nabila for the wonderful talk, very thought-provoking, fantastic work. Thank you, Edward and company, for helping with the logistics. It's a pleasure to be here. Let me share my screen, and hopefully this should work. I am an experimentalist after all, so do you see my cursor and my slides? Anyone? Can I get a thumbs up?
Yes, we do.
Yeah, wonderful. So lovely to be with you. We'll talk about the interface between electronics and the brain, and I will motivate why we want to do this and what are the wonderful things that we can achieve should we get this interface working. But before that, I need to get my presentation working. So let me share the screen again. This is a little hiccup that unfortunately does happen with Zoom. Let's see, here we are. Slides up. Presentation mode. So now it should go. Wonderful. So the motivation lies in the fact that the leading cause of disability is neurological disorders. According to a study that was completed a couple of years ago, there are over 270 million years lost to disability, a tremendous loss of productivity, of happiness, with stroke leading the way, migraine, dementias, and then epilepsies and other neurological conditions following. This is obviously a major societal problem. How are we going to solve it? How are we going to address it? Unfortunately, the drug industry is not going to help us. There is this trend which has been baptized Irum's law, which Irum is Moore spelled backwards. Moore's law in electronics says that things get faster and cheaper with time. In the pharmaceutical industry, it's quite the opposite. The amount of money that is required to get one drug approved keeps increasing. And as a result, the number of drugs that we can pass through the regulatory hurdles and get to the clinic decreases as a function of time, as shown here. And yes, this is adjusted for inflation. So it costs today over $1 billion to get a drug from the lab to the clinic. And the trend is pointing to a situation where things are going to get worse before they get any better. So the pharmaceutical industry is not going to solve this problem with neurological disease. However, there is good news in what is called bioelectronic medicine. This is truly a game-changing technology that involves electronics interfacing with the nervous system and trying to help it restore its function. Here is one example. This is called deep brain stimulation, and it involves implanting electrodes deep into the brain in a structure called the subthalamic nucleus. And through constant electric stimulation at a frequency of about 100 hertz, with a flick of a switch, one can help patients with motor disorders such as dystonia, Parkinson's, tremor, and epilepsy. And this is shown here. The system is implanted but turned off here. And the tremor, as you can see, is quite important. While here, with a flick of a switch, the tremor goes away to a large extent, allowing this patient to have a drink of water, as shown here. Now, this type of technology is also being tried for neuropsychiatric disorders, such as obsessive compulsive disorder, depression, as well as for conditions such as obesity. So very promising. And this goes back to the pacemaker, which was developed in the late 50s, early 60s, and started really this trend of implanting electrical stimulators to help our body recover from disease. So cardiac pacemakers currently are placed to the tune of about 600,000 patients per year. It's a very routine operation. Cochlear implants came about a decade later. These are for helping folks with hearing loss, severe hearing loss. And they're implanted to the tune of 35,000 devices per year. Then spinal cord stimulators, again, a decade past the cochlear implants, similar numbers in terms of patients benefiting from those. These are fitted for helping people with neuropathic pain. Then vagus nerve stimulation for epilepsy, sacral nerve stimulation for bladder control. Then the brain became a big target with deep brain stimulation for Parkinson's, for OCD. And then more and more devices were FDA approved. This is not a comprehensive list. The trend really appears to be exponential, a very rapid growth in the number of devices that become available and the number of conditions that can be treated with this. Obviously, this has created quite a lot of excitement and high expectations. There is commercial interest from entities that you wouldn't associate with bioelectronics, such as, for example, Google's parent company called Alphabet, that formed an alliance with a pharmaceutical to launch stimulators that are supposed to treat disease as alternatives to drugs. And then the founder of Tesla and SpaceX, Elon Musk, he also got into the game, and the founder of Facebook. So it's something that is very contemporary, excites people, and creates high expectations. And of course, that can be a bit dangerous, because if it doesn't deliver, then there will be a long winter. So what are the limitations? And they're pretty formidable. First and foremost, we do not understand how the brain works. Yes, these stimulators do have spectacular results, but how do they really work? The human brain is an exceptionally complex machine. There are over 80 billion neurons that are connected in networks and networks of networks. And although we understand how one neuron communicates with another, and this takes place through the propagation of an action potential, as it's called, which is pretty much like a voltage pulse that travels down the axon of one neuron, arrives at the synapse, and then through the secretion of neurotransmitter here at the synapse, it either excites or inhibits the postsynaptic neuron. This is something that we understand reasonably well in a quantitative fashion. However, how do we go from this, from the communication of two neurons, to behavior on the organism level is a long way ahead, and we do not understand that. We also do not understand what happens when we neuromodulate, what happens when we apply an electric current through a pair of electrodes. How do we change the behavior of neurons, and how do we change the resulting behavior of the organism? Now, in the clinic, we study the brain, by using different electrode configurations. For example, electrodes that are placed on the scalp in what is called electroencephalography. Electrodes that are placed on the cortex. Here you would need to do a craniotomy, remove part of the scalp, and then place electrodes on the surface of the brain without penetrating it. Or even in the most extreme cases, stereotaxic electroencephalography, where we introduce implantable electrodes that do penetrate into the brain and obviously are highly invasive. There is a trade-off between spatial resolution, how well you can record the brain and with what resolution, and invasiveness. This technique that involves implantable electrodes gives you the most precise information. You can get single neuron activity. You can listen to individual neurons if you place your probes appropriately and if they have a small size, small being of the order of 20 microns, which is the typical size of the soma of the body of a neuron. Human hair is roughly 50 microns, just for calibration. So this is obviously great in resolution, but unfortunately it's highly invasive and it's only reserved for extreme cases. At the other end, electroencephalography, you just stick electrodes on the skin. They can be removed very easily after the examination. But unfortunately, because you're sitting far away from the brain, of the order of a centimeter in a human, then you record very averaged out information. You record the chatter of many networks of neurons. Between the two lies electrocorticography, where you do not penetrate the brain. It's not as invasive as depth probes shown here. On the other hand, it was thought that the resolution is poor. You cannot get single units. You do a bit better than electroencephalography, but not as good. So anyway, with these techniques, people are trying to understand how the brain works and to understand what is the mechanism of neuromodulation. If you were to send a current through the brain, what would happen to behavior? These grand challenges, the kind of fundamental questions, go hand in hand with technological limitations that are very severe. The signals that are recorded, especially if you're at the surface of the brain or out of the brain, are very small. And you need quite good electronics to capture them. The environment in vivo, when you go into the brain behind the skin, is hostile to electronics. For example, silicon, which is the king of electronics, is the most traditional material that we know of and we make electronic circuits out of. If you put a bit of silicon in the brain, it is going to get etched, it is going to get oxidized and then hydrolyzed at the tune of about a micron a month. So our brain will just corrode and dissolve away the silicon and remove it. Very hostile environment. Moreover, the placement of these devices is highly invasive, both to the device and to, more importantly, to the brain. And often multiple surgeries are required in order to replace batteries and corroded leads, and this is certainly a major limitation. So this technology and fundamentals go hand in hand, and if we improve technology, then we could address the fundamental limitations and move bioelectronics medicine forward. So today I want to, with this introduction, I want to talk to you about three pieces of work for which we're very excited. The first one is how to teach electronics a foreign language, and I will explain why we need to do this. But the technical context here is we use materials that are called mixed electronic ionic conductors, and I will explain what that means, to communicate better with neurons in the brain. The second thing is that getting drugs into the brain turns out to be very difficult. In addition to the trouble of designing new drugs, getting it into the brain poses yet another hurdle. So I will show you how electronics can help with that. And finally, a very recent project for which we're very excited is implants that change shape. And as a result, they can decrease the invasiveness of neurosurgery. So another way to frame the field of bioelectronics is to consider the properties in the two worlds we're trying to interface. In living systems, the components, the tissues, especially the brain, are mechanically soft. The brain is like jello, very, very squishy and soft, while electronics in their traditional form are made out of mechanically stiff materials. If you take your cell phone, it's very hard to bend it and flex it. The communication in the components in the world of biology is very complex and often not well understood. So the way cells or tissues or organs communicate with each other involves a lot of messages that range from small metal ions, the sodium, potassium, all the way to fairly large chunks of cells that can be exchanged between one cell and another. While in electronics, the communication of these two components in this circuit takes place by an exchange of electrons, which is something that we understand on a quantitative basis. Biological systems evolve, they change shape, they change chemical composition. The brain of a teenager is very different than the brain of an elderly person. While electronics are static, the way we fabricate them is the way they spend, the form in which they are fabricated is the form in which they basically get recycled at the end of their life. So bioelectronics is all about building bridges and connecting properties. Now, in the last decade, there has been tremendous progress in making electronics in form factors that are similar to biology as far as mechanical properties are concerned. So we have electronics now that are very thin, very flexible, and they can be placed, for example, on top of the brain, forming a perfect mechanical contact that does not load the brain, does not cause any mechanical load. So the bridge, if you wish, between the two worlds in terms of mechanical properties has been built, and I will show you one example. So there is a lot of action today in getting electronics to communicate with complex signals. So going beyond just an exchange of electrons to understanding the language of ions and the language of more complex biomolecules. So this is what I mean by saying that we're going to teach electronics a foreign language. And this is used, for example, in the context of epilepsy, where you would use implantable electrodes that penetrate into the brain in order to record activities at different parts of the brain and localize the epileptogenic zone. This is a procedure that happens in patients who are resistant to anti-epileptic drugs and suffer from severe forms of epilepsy. In these cases, there is this explorative neurosurgery that takes place in order to localize which parts of the brain give rise to an epileptic seizure. And then a decision is taken to remove that part of the brain, the so-called epileptogenic zone, and cure the patient. So here again, there are electrodes at different parts of the brain, and you see here the manifestation of a seizure. It really looks like an earthquake on a seismogram, a very high amplitude activity. And what the neurologists try to do is to extrapolate and figure out where does this activity originate from. Did it originate from this brain area, or was it this electrode here in another area of the brain that captured this activity? Was that a prelude to triggering the seizure? In order to take these decisions, you need electrodes that have superb performance, that couple very well with neuronal activity. And now, for a little bit of material science, the way you would do this is you would try to interface ionic signals, signals that are arising from the motion of ions such as sodium, potassium, inside the brain, in the electrolyte that exists inside our brain, and it's called the cerebrospinal fluid. You would try to make these ions interact with electronic charges, electrons or holes, inside your electrode. If you use silicon, again, the king of electronic materials, you would run into the issue that you would need to protect your chunk of silicon with either an oxide or a nitride. So this layer spaces away the ions and the electronic charge at the two sides of the interface and decreases their coupling. So the communication between ionics, the brain, and electronics, the electronic material, is just not very strong enough. Even if you could remove this oxide layer, use, for example, a noble metal such as platinum, you would arrive at a situation where you would have ions on one side of the interface in the brain, and then electrons on the other side of the interface in your electrode, in your platinum electrode. And this coupling along the interface is fairly weak, and it's characterized by what is called the double layer capacitance. It's a fixed value, and it just limits to how close these two carriers, how strongly these two carriers can interact with each other. On the other hand, if you use a material in which ions can penetrate into its volume, and this would be a so-called conducting polymer. We're going to overlook chemical details here. Just consider this as a spongy-like material in which ions can penetrate. Then you would have coupling in the whole volume of the material. So ions and electronic charge will couple much more strongly. So you increase, in a sense, the communication strength between brain and electronics. So when neurons talk and put these ions in motion, when they talk to each other and set these ions in motion, your electronic material can listen to this with a very strong coupling, very strong efficiency. So this simple concept allows you to make novel devices that have state-of-the-art performance. Last slide with chemistry, I promise. So here are two materials that look very similar. The only difference is that here you have a wiggly side chain. This is an alkyl side chain. And here on the same wiggly side chain, you have oxygen atoms. This tiny modification in the chemical nature of the material has endows major changes in performance. In this material, ions will not penetrate in the volume, will stay on the liquid side of the interface, will stay in the brain. And as a result, the coupling between ionics and electronics is very weak and only takes place on the surface of the material. So if you were to measure the so-called capacitance, which describes the strength of the interaction, it would be the same as that of a flat metal film. So a two-dimensional interaction, which is weak. On the other hand, in this material, the addition of oxygen atoms on the side chain allows ions to penetrate into the volume. And then all of a sudden, the capacitance increases quite a lot. So the strength of communication increases. Turns out there is an ideal material for this purpose that has perfect volumetric conduction. Ions can go into the volume and couple very efficiently with electronic charge. And the way this manifests itself is in a capacitance that scales with the volume of the material. The larger you make your electrode, or the thicker you make your electrode, the higher the capacitance. What does that mean? That means that if you just coat a metal electrode with a film of this polymer that is just 130 nanometers thick, a very, very thin material, you're going to have an increase in the coupling between ionics and electronics of 2 orders of magnitude. So a magical material, you just put a wafer thin coating on your electrode and you have enhanced communication with the brain 100-fold. This means that now you can make small contacts, you can make 100 times smaller electrodes and still be able to record with the same accuracy, with the same signal to noise ratio as a much, much larger gold electrode or platinum electrode. So you just gained in spatial resolution. So we played this trick. We coated gold electrodes with this polymer layer and introduced these electrodes on a platform, on a plastic foil, which is very thin and very flexible. It can actually follow very tight radii of curvatures. Here is shown on an orchid leaf, following radii of curvatures that would be consistent with getting into sulci in the human brain, and provides negligible mechanical loading on the brain. It has very small electrodes that are capable of recording with exceptionally high performance. And when this was tried on the cortex of rats, we saw this spikes shown here in the recording that signify that we can listen to individual neurons, the chatter of individual neurons. This was a really interesting finding because people believe that from the cortex, from the surface of the cortex, it would be impossible to listen to single neurons because the signals that emanate at the surface of the cortex are very weak and require very large electrodes to capture. By using this polymer layer, we're able to make smaller electrodes that can capture these very weak signals and then focus on single neurons and listen to their chatter. This was an overwhelming advantage compared to established technologies, and it made it to the clinic in record high time. So it was first developed in 2011, and by 2015, it was already used in hospitals to map the human brain of epileptic patients with exceptionally high resolution, as shown here. So these arrays do measure the macroscopic activity that's called local field potentials, and it consists of the chatter of many, many neurons in the neighborhood of the electrode. But they also measure these little spikes that consist of individual neurons right underneath the electrode. Now, it turns out you can do a bit better by using electrodes, by using transistors. And I'll very briefly talk about this. The argument goes as follows. If you have the brain and you're recording its activity with an electrode, you're recording the signal that arises from the brain, which is the activity you want to record. But there's also a lot of biological noise. There are other activities that go on in our head, which is not what we're looking for. And this is called biological noise. So you record all that. Then you need to have your electrode connected with a wire to an amplifier and a recording signal. And this cable acts as an antenna that picks up signals from cell towers, the lights in the room, the anesthesia equipment. So you end up having quite a lot of noise. And then when your amplifier amplifies this, it amplifies not only the signal, but also the noise. And the argument goes that, let me run through the slide here, that if you were to move your amplifier close to the source of signal. You would amplify signal and biological noise, but not amplify any additional noise that is picked up on the line. So you'll end up having a better performance, better signal to noise ratio. So this motivated people like Peter Fromhertz at the Max Planck Institute to develop dedicate their career to the development of transistors that would record neuronal signals with high amplification locally, right at the interface. So again, the configuration is fairly similar to that of electrodes. You have your silicon here, you have an oxide or a nitride that separates the electronics from the cell culture media in this case. And you would have an ionic layer of charge coupling with an electronic layer of charge inside your transistor channel, as it's called. And when the neuron fires, it would change this ionic charge. That would change the electronic charge in the silicon, and you'd be able to detect it. But again, as I mentioned earlier, this two-dimensional coupling between ions and electrons that's mediated by an oxide is very weak. In the 80s, 84, Mark Crichton, at that time at MIT, developed a new type of transistor, which uses organic materials, such as the conducting polymer I showed you earlier. And in this case, the polymer layer is in direct contact with the electrolyte. So ions can go into it and change in the whole volume its conductivity. And turns out with this trick, you can make transistors that operate at least 100 times better than state-of-the-art silicon transistors of the same dimensions. I'm not gonna go through the details of these curves. This black curve here shows the sensitivity of the device, how much change you have in your current, which is your output, every time you have a voltage because of neuronal activity. And by utilizing this volumetric conduction of ions in polymers, you can get exceptional performance. And that then allows you to record the brain with much higher performance. These are cortical arrays that are sitting on the surface of the brain of a rat, which is a model for epilepsy. And they have both transistors here and electrodes. And the electrodes are at the state of the art. They do have this polymer layer that operates exceptionally well. This is the recording of a series of seizures with the electrode. And this is the recording with the transistors. And you don't need to do any particular analysis to convince yourself that this contains more information than that. So it speeds up the pace of experimental research. Not only that, but it allows you to see deeper in the brain from the surface. These are 3 recording configurations. Here we have an electrode that penetrates into the brain and comes very close to the origin of neural activity we're trying to detect. So this is the signal that is being recorded, and this is what is called the time frequency analysis that shows the frequency components of the signal as it's being recorded. And then this is the same signal, but recorded not with a penetrating electrode, but an electrode that sits at the surface of the brain. And as such, it shows fairly weak resolution of the signal. It just doesn't capture it in the same way that is being captured here. But the transistor, with the amplification that it provides, recuperates quite a lot of the lost signal. So it can be used to also look deeper into the brain than an electrode. And let me stop here and move on. So what I talked about so far was devices that use materials in which ions can penetrate and couple very strongly with electronic carriers as a means to record better from the brain. Now we're going to turn this around, and we're going to use similar materials to send ions into the brain for the purposes of drug delivery. And the application here is, again, in the context of epilepsy. As I mentioned earlier, the first line of defense against epilepsy are anti-epileptic drugs. Unfortunately, the 30% of patients do not respond to these drugs for reasons that are not known. In this case, and when epilepsy is severe enough to be disabling, resective surgery is the next line of treatment. So in this case, the region of the brain that gives rise to seizures is localized with exploratory neurosurgery, as I showed earlier, and then it's removed by resection, and the patient is cured. However, this resection is not always possible because sometimes the epileptogenic zone overlaps with an area of the brain that has high functionality. And then the trade-off between benefit and risk to the patient would just not pan out, or gain and loss of performance. In these cases, we advocate implanting a device in the epileptogenic zone, leaving the brain undisturbed, and effectuating localized delivery of drug when and where is needed. So where is needed is in the epileptogenic zone, and when is needed, it would be just before a seizure. Now, this local drug delivery is a very interesting proposition because the brain has the so-called blood-brain barrier. It's a barrier that protects and blocks the transmission of drugs from the bloodstream into the brain. About 98% of drugs that we have developed will not cross into the brain with high efficiency. An implantable device will already be past the blood-brain barrier. So all of a sudden, you have a much larger gamut of drugs that you can use. In addition, you can use drugs that are very powerful and would otherwise be toxic to the body, but because you're only dosing them locally at minute concentrations, you can use them. You're able to use them. So this proposition of local drug delivery past the blood-brain barrier, it's something that has attracted quite a lot of work. And this is one example from the work on cancer, where after resection, you want to locally deliver some chemotherapy to kill the remaining cancer cells. And this is done through a couple of different configurations. The most promising is a catheter that where you apply pressure and release the solution of a drug locally into the brain. However, these concepts would not work in the case of epilepsy. They quite frankly, they don't work well in the case of cancer as well, partly because in order to supply the drug, you would need to apply quite a lot of pressure. The brain is enclosed by the skull, and that pressure causes quite a lot of damage. And also lots of the drug just comes out through the side of the catheter. So we were very excited when about a decade ago, we saw a device called the organic electronic ion pump that was developed by a group in Sweden, that uses materials very similar to the ones we use for electrodes to transport ions from one reservoir to the other. So what you do here is you have a film of this material, you apply a potential, this film bridges two ions, two liquid reservoirs, and the application of a potential makes it so that you get ions from one reservoir to transport and diffuse into the other. The Swedes use this to demonstrate the delivery of a neurotransmitter in the ear of a guinea pig as a way to study audition. The way this device works is it has a membrane that is called an ion exchange membrane separating two reservoirs. This is the version that of the device that we use. One is the source reservoir that has your drug, which is in cationic form in our case. And the membrane separates that reservoir from the brain. Now, the membrane is made out of a polymer that has negative charge that is fixed to the polymer structure and cannot move, and also has positive ionic charge that is able to move about. So when you apply a potential between the inside and the outside of this device, you would have the motion of the drug from your source reservoir through the membrane into the brain. But at the same time, the opposite ion, negative ions, will be blocked, will not be able to go through this membrane because of all this fixed negative charge. So it doesn't subtract ions from the brain, it just adds the right ion into the target site of the brain. So this is how a device would look like in real life. This is again for rodents, and you would have a U-shaped microfluidic channel that brings the drug into the device, into the brain. This is the drug solution. And then there is a U-turn here, and there are holes poked in the microfluidic channel and covered with this ion exchange membrane. So by applying a potential between the inside and the outside of the device, we can get just the drug and not the solvent to come out and be delivered in this area of the brain. And the solvent, the depleted solvent, just takes a U-turn and comes out of the device. So this particular device doesn't increase the pressure in the area of the brain that it delivers the drug, because it doesn't deliver any fluid. It only delivers the drug molecules when they're used. We were able to show that we can deliver GABA. GABA is the inhibitory neurotransmitter that is native to the brain and represents the brake signal for neurons to stop firing. And this is a device in the brain, and just by applying half a volt, we can increase the local concentration of GABA by several orders of magnitude and achieve a therapeutic effect. This is shown here in, again, an animal model for epilepsy. In scenario A, the device is implanted, but it's turned off. And we kindle a seizure, and you can see the seizure manifest itself. They become more frequent and more intense. Here, scenario B, the seizure is kindled, and as it appears, we turn the device on and we start delivering GABA. And the spikes that you see here correspond to an electrical artifact that appears every time we puff a little bit of GABA into the brain. But the thing that you see is that the seizure immediately disappears after applying GABA. And then in scenario C is we prophylactically turn the device on. You see again the spikes that correspond to puffing GABA into the brain, and then try to kindle the seizure, and no seizure manifests itself. So it can be used not only to stop, but also prevent a seizure from arising. Very interesting results. The road to the clinic is long. These have to go to more testing in larger animals that are inherently epileptic, genetically susceptible to seizures, on their way to eventually the clinic. Now, one thing that you learn very quickly in this game is that one device doesn't fit all situations. For example, local delivery is very interesting for epilepsy. It's also very interesting for brain tumors. But the requirements for a treatment in this and that condition are very different. For example, the target volume in epilepsy is very small. Typically, it can be as small as a single cell malfunctioning and causing seizures. while brain tumors, typically by the time they're discovered, they're centimeter size. So we spent quite a lot of time modeling these devices, trying to understand how they work. And it's not surprising that you find that if you look at regions, these are two parameters that we can easily tune. And to maximize the amount of drug being transported, you need to be here. To maximize the on-off ratio, how much drug you have when you turn the device on versus how much passive leakage of the drug you have, this is optimized in another parameter regime. So for every type of treatment, for every disease, we have to re-engineer the device appropriately. But it's very interesting that we can make these devices deliver at very large voxels. This is, again, a brain tumor, and it can be massive, several centimeters in size. And these devices can be engineered to address such large volumes. This is in the context of an EPSRC project. And we hope that this device will be in the clinic for brain cancer within about 6 years. We're trying to push this as far as and as fast as we can. Now, in the last 3 minutes, let me show you some recent data. I showed you this slide before, talked about the mechanical properties mismatch, talked about the communication mismatch. What do we do about the dynamic versus static mismatch of properties in the two worlds? And a way to frame this is to state that neurosurgery is very invasive. So this is the placement of a cortical probe, a commercial cortical probe, and requires a craniotomy, removing part of the skull and opening the dura in order to slide this device in. Wouldn't it be wonderful to just open a small burr hole on the skull, put in a device in the form of a needle, and then deploy it without having to remove a big piece of the brain? Similarly, for the placement of spinal cord implants, a laminectomy is usually performed, which is quite brutal surgery. While in principle, you could have a device that would be small, inserted through a lumbar puncture, and then change shape, be inflated or deployed in order to cover the spinal cord. So this, in a sense, called for coupling bioelectronics, the way we make thin and flexible electrode arrays with soft robotics, structures that use fluidic activation to achieve mechanical motion. And with this, we were able to make arrays that can deploy either in a forward fashion, as shown here, be rolled and then deploy into a linear long array, or rolled sideways, as shown here, and deploy into a paddle electrode. So this is a typical electrode that is used for spinal cord implantation. This is for patients with neuropathic pain. And again, it requires a laminectomy to place. While you can roll it, if it's made appropriately, into the shape of a needle, it can be inserted through a lumbar puncture and then deployed as shown here. This is an in vitro model of the spinal cord. So it can be inserted as a needle and then deployed when needed. This we've seen in human cadavers. You can do this in the spinal cord of a human cadaver and deploy such structures, but also in the brain. This is a cortical implant that has been implanted through a small burr hole and then deployed over 2 centimeters. And these are tracers that show the edges of the implant. So this provides a lot of hope for being able to address the invasiveness of neural surgery through implants that change shape. So with that, I gave you a couple of examples, and I hope above and beyond that I managed to convey my excitement about implantable electronics. I really believe that they hold considerable promise for helping understand the brain, and address its pathologies. We talked about electrodes, we talked about drug delivery devices, and about devices that change shape as a way to minimize invasiveness. So with that, let me thank my group, collaborators in Cambridge, outside Cambridge, and funding. A colleague of mine ends his talks by saying, we all have a vision, scientific vision, but without funding, it's just a hallucination. So we thank the funding agencies for helping us keep it vision and not hallucination. So thank you for the invitation. Thank you for your attention. I welcome your questions.
Well, Professor Malliaras, that was absolutely spellbinding. You could hear a pin drop, even if it was a full room. Well, I can see one or two questions coming up if you're ready to roll, Professor Malliaras.
Yes.
And can I just say to people, I mentioned before that you can ask a question on the Q&A, but if you would like to go on video, there's a button you could press to raise your hand. And if you would like us to see you, then press the raise your hand button, and Edward will get in touch with you and we'll get you on the telly. But first of all, I have a question from WS. Is it Mr. Mrs. Miss Steele or W Steele? I have a report. I saw a report a couple of years ago where an electronic implant allowed a blind woman to see shades. How close do you think we are to something similar to the cochlear implant, but for vision?
Yes. So the technology, it's very similar. The main difference is that the cochlear implant can function reasonably well with a dozen electrodes. A retinal prosthesis device requires more than 1,000 electrodes in order to begin functioning in a reasonable fashion. There are a couple of devices out there that are approved by the FDA. The Argus 2 implant is one example. I believe it has less than 1,000 contacts. It allows patients to read large letters to the tune of about a letter a minute. So it provides very, very limited vision, but it's an engineering problem. It's we need to reduce power consumption. We need to make the electrodes smaller and maintain their efficiency. We need to figure out better ways to couple power from outside the body into the implant. We need better ways to process images before we send them to the implant. There is clearly a path that will lead to progress. And also there are some disruptive concepts. There are now, instead of having a full implant that consists of a camera, a computer, and then an electrode array that's implanted, there are just materials approaches where you would have a tri-layer film, a structure that is implanted in the retina, and it's the same structure that absorbs light that goes through the eye and triggers the remaining cells in the retina. It's always very difficult to state a time scale for progress. So I actually have a friend who is looking into getting these devices, and it's very difficult to tell when the next generation will be available, whether it's incremental or it's really worth the effort in getting it. There will be progress, but how fast and how much difference it will make in patients is hard to tell.
Understood. Thank you. And I believe we have a video question from John Cook.
Hello, Professor Malliaras. That was a fantastic talk. I had really no idea we were doing anything like this. While you were talking about the electrically induced production of drugs for epilepsy into very small parts of the brain, I started wondering, could this be done autonomously, do you think, where you have a device that detects the onset of a seizure, and you have a reservoir of the drug, and the device decides itself? You'd need a power supply, but you know, we can do this kind of thing. Is that on your horizon somewhere?
Yes. This such closed loop systems where you detect, you decide, and then you actuate, are being looked at very seriously. For example, in Parkinson's, in deep brain stimulators for Parkinson's, there are adaptive models that would feed into, for example, tremor. The patient would wear a ring that would have a little actuator and would measure the frequency of the tremor. And that information is conveyed into the implant that would then adjust its performance. The key aspect here is to find the appropriate biomarker, to find an activity that can be measured precisely and necessitates a change in treatment. For example, in the case of epilepsy, as you mentioned, you would need to be able to detect an oncoming seizure and then activate the pump with enough time to stop the seizure before it manifests itself. There are electrical biomarkers. By looking at electrical recordings of the brain, you could predict an oncoming seizure with decent accuracy. We are looking into biochemical markers. We're looking into changes in local metabolic activity in the epileptogenic zone as a means of predicting a seizure accurately and then triggering the pump. We have done something along these lines, but using an external system where the operator is pressing the button when it's seeing a trend on lactate. But in principle, down the road, we could envision such systems.
Fascinating. Professor Malliaras, Amy is asking whether it's possible to access the slides. I know sometimes these things are confidential.
Yeah, no, please send me an email and I'll be happy to send the slides.
That's great, thank you. So perhaps we'll pick them up at the office end, Amy, and we can get them through to you. I've got another question. By the way, if anybody would like to put their hand up, we'd be pleased to see it. Press the button if you would like to be on the telly. But meanwhile, Sean Blake says, have these techniques application the control of prosthetic limbs by capturing cortical signals or signals from peripheral nerve trunks?
Yes, there are demonstrations, very powerful demonstrations out there. Most of these demonstrations require quite a lot of equipment. So it's not out of the lab yet. It requires quite a lot of support to get the implant working in a stable fashion and calibrate and all that. So as you said, it would be an implant on the motor cortex, reading volition. When the patient thinks, I would like to move my left arm, the neural signals that correspond to that activity can be read by the implant. Then a computer would interpret those systems, and that's those signals, and that's the part that needs quite careful calibration and drive a prosthetic or an exoskeleton. It is demonstrated in primates, in humans, a lot of work in the US funded by DARPA, but it's not widely deployed yet. It's expensive. There is a project in France that aims to democratize, as the French would say, this type of prosthesis, make it more mass-produced and decrease the cost, but it's still early days.
Thank you. And now we have a question from Paul Klein from Anglia Ruskin University. How do you envisage this technology assisting OCD? Some kind of neurodampening, perhaps?
Yes, I don't know, and I don't think anybody does. It's the mechanism of neuromodulation, especially when it comes to neuropsychiatric disorders. It's absolutely not understood. That's a very, very good question. Usually, the way it works for Parkinson's, for example, how did they think to stick an electrode and stimulate the brain as a way to stop tremor in Parkinson's? The way it happened is a particular structure inside the brain has been identified through a lot of previous work as a target for stopping tremor. And they found that if you remove that part of the brain, if you damage it, if you resect it, then tremor improves. And then there was an accidental discovery. They found that they were trying to burn that part of the brain away by using an electrode and passing locally high current. And the hypothesis was, rather than putting a knife and cutting it, we'll burn it. But well before it started to cook, as they were raising the current, the tremor stopped without damaging that part of the brain. And obviously this is desirable, right? You do much less harm. So that was the birth of DBS for Parkinson's. Sometimes there is an accidental discovery, but it relies on a lot of previous work to identify the target.
Great, thank you. And I can see John Scott on the telly, if you'd like to give us your question, Scott.
Hello, thank you. Fascinating talk. Going back to the epilepsy question, is it well known how to detect the onset of epilepsy, and is it a seizure? And is it also known how precisely you have to provide the treatment? So in other words, positioning your electrode for introducing a drug or whatever, is that very consistent between, for a given person, say, over their lifetime of having seizures? And the other thing that did occur to me is, I did read somewhere recently that apparently dogs can be trained to predict epileptic fits in their owners. And does that give another opening as to an input into firing the pump or the treatment, rather than picking up electrical signals on the brain?
Yeah, these are very good questions, and some aspects are beyond my expertise. What happens is, in cases where epilepsy is severe and intractable, there is this exploratory neurosurgery that aims to localize the epileptogenic zone. So there is a lot of preparatory work before that points a little bit to some target brain structures. For example, based on the symptoms that the patient displays, the neurosurgeons can make an educated guess as to the approximate location of the epileptogenic zone. Then they would implant electrodes there and keep the patient in the clinic for a period of 2 weeks to accumulate enough seizures, enough recordings in order to be able to localize. And they would localize with precisions that are of the order of, if you consider it as a voxel, of the order of typically neural surgery has about 1 millimeter precision. Now, in some patients, so in about 30% of patients who are candidates for resective surgery is not performed. The most common reasons is that the epileptogenic zone overlaps with an area of high functionality, or there are multiple epileptogenic zones, or they cannot localize the epileptogenic zone altogether. I don't know how it breaks down in these three categories, but overall, about 30% of cases, there will not be a resection. There are examples where the epileptogenic zone can be stimulated to prevent seizures or stop seizures, but then it would move, or it would it would become, there would be a phenomenon of habituation where the treatment would become less effective in time. So all these are very much up for question. Is not there are no clear answers in a lot of the questions. Yeah. Welcome.
Thank you.
Well, Professor Malliaras, I think we should allow you to let your family escape from lockdown, which they've probably been in for the last well over an hour. Thank you for getting our season off to an absolutely splendid start. I think much of what you've said will have been quite revelatory to many of us. We had no idea that this kind of thing was possible. And of course, the implications are profound. You've already ranged across all sorts of aspects of life where this technology can be influential. So can I thank you most warmly for an absolutely lucid but very complicated presentation, and for answering all these questions in such an authoritative way. So our warmest thanks to you, and I hope in due course to be able to bump into you in the engineering department.
I look forward
Thank you very much indeed.
Thank you for your invitation. Thank you.